Thank you, Martin, and Dober Chair, everyone. Martin, congratulations to you and the team for putting together a really great conference.
Thank you.
No technical hitches whatsoever, which is amazing. And it makes me question why scientists have been tramping all over the world to go to these scientific conferences when we can do this by Zoom. And that, you know, with the exception of some face-to-face interactions and maybe spending some time at the bar together, you know, this is almost as good as being there in person. So congratulations for that. And thanks to Aubrey. I haven't seen him for a few years now, and it's always tough to follow him up because he has so many great ideas and he thinks about these things in so much detail. So I'm Stephen Minger, and I'm a neurobiologist and stem cell researcher. I'm going to take a completely, or at least probably quite different approach than most of the previous speakers. And I'm really going to address longevity in any great extent specifically, but what I am going to talk about as a technologist who, as the name of my company implies, Blue Skies, is constantly looking to the future, new emergent technologies, which, at least in my estimation, humble estimation, I think will be transformative in terms of healthcare and aging and longevity, and you know, in a myriad of different ways. So let me start with what I'm most familiar with, which is, you know, stem cells. And as somebody who has a long interest in age-related diseases, particularly those of the nervous system, which is where I started out 30 years ago, back in California in the 90s, we discovered that there were stem cells, certainly in the developing brain. And then after a few more years, we discovered that there were stem cells in the adult human brain, which are actually, in principle, we think capable of quite extensive regeneration, although this is a really untapped area, and I'll come back to this later, but it really kind of fueled a stem cell revolution. And then, as you'll see, we moved beyond the nervous system, beyond stem cells in the brain, to a whole variety of different stem cells. And I think now, you know, after, in some cases, 40 or 50 years of working with what we call adult tissue stem cells, and more recently, what we call pluripotent cells, and I'll make this distinction in a moment. You know, if you go into clinicaltrials.gov, which is the major database for clinical trials in any clinical area, there are over 4,000 quote unquote stem cell clinical trials at the moment. The vast majority of them take advantage of two stem cell populations that we find in adult humans. Professor Roseman Primash has already talked about one of these, the one at the top, which are the stem cells, the very, very rare but very powerful hematopoietic stem cells, which drive the differentiation of cells into all the myriad cells in the vasculature, red blood cells, white blood cells, macrophages, et cetera, et cetera. These cells have been really a workhorse of hematology for over 50, 60 years now, as Primash talked about in terms of bone marrow transplantation. Starting in the 1970s, probably more in the 1980s, a second population shown down here at the bottom that also reside predominantly in bone marrow in adult humans, what we call mesenchymal stem cells, mesenchymal stromal cells. They have a variety of different names. Their exact lineage and their exact definition still remains to be determined. But nevertheless, these cells are probably found in all tissues, not just in bone marrow, but that's traditionally where we've isolated them from. And these cells are unique in the sense that they probably are the cells that give rise to bone and to cartilage and, to some cases, fat in our bodies, and have some regenerative capability as we age. We repair bone, we put down new bone, but this, as Aubrey and others have pointed out, this fails over time, and our stem cell populations just are not as robust as they were when we were 70, 80, 90 than when we were in our 20s and 30s. But I think more importantly is we're discovering these cells are actually really, and this has only come in the last 5 to 10 years, these cells are actually factories that secrete a number of important molecules which have profound impact on regeneration, inflammation, modulating the immune system. There are clinical trials here in Ljubljana, in Slovenia, on using MSCs to dampen down a nasty disease that comes from organ and bone marrow transplantation called graft versus host disease, where there's a mismatch in the immune system between the host and the donor. This oftentimes results in death. It's not treated very well, and when it's severe, quite often the patients die. We've shown, not myself, my colleagues have shown, Miomir, Knisevich, and Educel have shown that these cells are very capable of reducing this immune response that comes from the new donor cells going into the host. These cells were also used very recently in patients with COVID-19 induced acute respiratory distress syndrome. This is when these really severely sick patients with lung inflammation have to go on ventilation. A lot of them die, and the ones that survive will have greatly scarred lung tissue. And in data, idiosyncratic data from patients in Italy and in Serbia, we've seen that there's a dramatic reduction in the inflammatory markers in the blood and in the lungs of these patients. So it may be that these cells are capable of homing to the inflammation, secreting appropriate factors that reduce respiratory response, and perhaps repairing some lung function. The problem is by the time patients go on ventilation, their lungs are really, really in desperate straits. And so this is really treating end-stage patients, which is not what we like to do. Nevertheless, it gives you some idea of the power of these cells. So I'll come back to this in a little bit later, but these cells are being used in a myriad of different clinical trials where we think inflammation, recruitment of quote unquote regenerative cells from other tissues to repair tissues, dampen down inflammatory responses is another way that these cells are useful over and above building replacement cartilage and bone. The work that we're most known for, I guess, is work that we did many years ago on isolating cells from the fetal brain. Aubrey talked about trying to make replacement neurons for Parkinson's disease. We have data from groups in Sweden and in the States that if you isolate the cells from a developing fetus, say at about 6 weeks, 7 weeks of gestational age, and implant those into the brains of Parkinson's patients, you can affect long-term sustained clinical improvement in patients with quite moderate Parkinson's disease who are already on drug and who are already quite impaired. If enough of these cells go into the brain, they survive and they integrate into the brain. Many of these patients stay off of dopaminergic pharmacotherapy and have long-term sustainable grafts for 25 years or longer. The problem is the cells are obtained from very early elective terminations. There are ethical issues, there are logistical issues with getting enough tissue. And so many years ago, we worked on this and we tried to find ways of being able to generate enough cells from even earlier embryos, say 4 weeks gestation, which, you know, logistically is almost impossible. And so we looked for alternative sources of cells that could give rise to large numbers of dopaminergic neurons. And we and others turned to embryos. And so this is a picture, a photomicrograph of a 6-day-old human embryo in a dish in culture. This is an embryo that was donated to our research program in London when I was at King's College London. This is a 6-day-old embryo created by in vitro fertilization. So a woman donates eggs, her partner donates sperm or a donor. Embryos are created for the purposes of fertility treatment, so to try to create babies. But due to the large number of embryos that are created and never used, in the UK, we had very strict guidelines for how these embryos could be donated for research. And we use them, and the important thing about an embryo at 6 days of gestation is that it's the first division into cells that, in the one hand down here, the inner cell mass, these cells will go on to create the embryo proper. And this larger population of cells, which is called the trophectoderm, is actually the primordial placenta. This embryo, were it in a woman, would still be about 3 days before implantation into the uterine wall. The unique property of these cells at this point in time is that they are pluripotent. That is, they can give rise to the entire embryo. And what Jamie Thompson in Wisconsin, Joseph Ishkovitz Elder, Israel, Arif Bangzo in Singapore, and then ultimately our group showed, is that when these cells are isolated from this developing blastocyst, put into culture, you can generate a cell line that's equivalent to that small population of cells that's in the blastocyst, and you can expand these cells ad infinitum. And to give you a sense of this, this is one of the first 3 cell lines that were created in the late 90s in Wisconsin by Joseph and Jamie, called H7. This cell line has been used by thousands of research groups worldwide. The numbers of cells that have come from this one embryo and 20 years worth of work in thousands of labs across the world, including my lab in London and my lab at GE Healthcare, would fill, I don't know, an 80-story tower in London, probably twice over. These cells are immortal in the way that cancer cells are, but at the same time, they're also genetically stable, they're normal. And the unique feature of these cells is that they can turn into every cell in the body, you know, because they have the capability within them, inherently within their DNA, and a developmental program, if exposed to the right factors, that can give rise to an entire baby. In that case, it's the signals they get from the uterine wall. In our case, it's chemical factors that we feed them in culture, and we try to drive them in different directions. So to give you an idea of how powerful this is, and in a moment I'll tell you where we're at clinically with this, and even probably as importantly in a research perspective, is, you know, the big challenge is, how do you take cells that want to turn into every cell type in the body, and you only turn it into the cells that you want, and you don't end up with these other cells that you would rather not have. And this has taken a long, long time. Even though many of us have been working on these cells for 20 years now, it's only recently that we've begun to really start to push them in very defined directions and turn them into highly specific sets of cells. So when I left academia to join GE Healthcare, one of the things that we wanted to do were to develop a protocol, for generating human cardiomyocytes, human heart cells. And at the time that we started this in 2009, I thought it would take us more than 5 years to do this. We actually figured out how to do it together with our colleagues at Geron Corporation in California, which was the leading human embryonic stem cell company in those days. And it's actually quite a simple procedure, about 30 days long. You start with completely undifferentiated human embryonic stem cells that can turn into everything. You force them to become the middle layer in a developing embryo, which is called mesoderm. Mesoderm forms ultimately will give rise to your lungs, heart, and all the vasculature, muscle. You then push those cells to become cardiac progenitor cells, and then you differentiate them into human adult heart cells. And this is what it looks like at the end of the process. We have at the scale that we were working with, we had 4,225 centimeters squared flasks covered with contracting myocardium. Every flask would do this. About 80% of the cells, if you analyze them in detail, either electrically, morphologically, or molecularly, are human cardiac cells. About 80% of them are what we call ventricular, that is, they're cells from the ventricle, just like your heart has ventricles. About 18 or 19% of them have an atrial phenotype, which is quite different. And those are the cells that form the atrium in your heart. And about 1 to 2% of the cells are nodal. They're the pacemaker cells that cause this whole thing to contract in synchrony. If you notice, they are beating together. They're not beating asynchronously and randomly. They are beating together. Routinely, we would make about 80% heart cells from each of our differentiations. You can see some underlying cells here in these holes. These are fibroblasts, which actually support the differentiation of these cells by secreting a lot of really interesting growth factors. We absolutely need them. If you were to try to do this with 100% heart cells, they don't survive. Now, surprisingly enough, we can take these populations, which are very mature, have an adult heartbeat. If you count the beats, it's roughly 70 beats a minute, like your heart. If these were fetal, it would be more like 120 or 140 beats a minute. You can dissociate these down to single cells, put them in liquid nitrogen, freeze them for 5 years, ship them to Australia. People take them out of liquid nitrogen, put them in the dish. Within 3 days, they reform this syncytia that I just showed you, and they spontaneously start to contract. So we're not driving these cells electrically. We're not stimulating them in any way. They are doing this all on themselves. They self-organize into a structure that is something like a primordial heart, okay? And this is data from us that's probably from about 2013. Just this week, 2 important papers have come out. One will come out in Nature tomorrow, and another one I think is in Cell. That is groups in scientists working with very similar to cells to us have come up with a protocol whereby they can create highly differentiated human lung cells in these, instead of the cells like I've showed you here, which are in a monolayer flat in a dish. A lot of groups now have gone to kind of organoids, these 3-dimensional structures which kind of float around rather than being adherent on the dish like these guys. And these lung organoids express phenotype very, very similar to human lung tissue, which is almost impossible to obtain except from biopsy. And even then, these cells really, because they come from adult tissues, they perform very, very poorly in culture. And what this one group has shown is that they have very, very good lung cells. And they're looking at actually how CV2, the coronavirus, which causes COVID-19, how it is so destructive in causing this ADRS syndrome in human lung patients. So we're developing not only the potential to use these cells in therapy, but we're now developing similar cells that are proving to be very, very useful in modeling human disease where we don't really have other available tissues. Fetal cells would have very, very different properties. Mouse and rat cells would have very, very different properties. And human cells, either obtained by biopsy or cadaveric tissue, would just be very, very poor. So when you look at where we're at, these cells are proving incredibly useful in modeling human disease as well as for therapy. I'll talk about the therapeutic aspects in a moment. I just want to make one other point for those of you who are not aficionados of this field. What I've shown you is we take a donated embryo blastocyst, we isolate that population of cells from the inner cell mass, we make a pluripotent cell line that we can drive into, we think, almost any cell type: lung tissue, liver, skin, heart, bone, muscle, etc. Not long after this field really was established, around 2003, 2004, predominantly in the UK. In the US at the time, it was very difficult to do this. A really brilliant chap named Shinya Yamanaka in Japan wondered about what's unique about these cells. How is it that these cells are so much different than even stem cells from the bone marrow, from the umbilical cord, from skin, from bone, etc. And Shinya did a genome screen on pluripotent embryonic stem cells and came up with a long list of genes that were highly overexpressed, which he thought might be what we call pluripotent genes. That is, genes that confer this pluripotent state on these cells, which are not expressed in other stem cell populations. And in a really heroic set of experiments, and the number of genes was around 50 at the beginning, they started putting these in sets into cells that were not even stem cells. They were basically fibroblasts obtained from mice. And over about 5 years, they found that there are actually only 4 genes in pluripotent stem cells that can confer pluripotency on a cell that has never, ever been a stem cell and is never going to be anything other than a stem cell. And when you overexpress these 4 genes, OCT4, SOX2, NANOG, the 4th one, there are about 3 or 4 different genes that can suffice, but you have to have OCT4, NANOG, and SOX2. You can make a population of cells that for the most part is indistinguishable from the cells that you make from an embryo. Why is this important? It's important because when we get cells from an embryo, they have the genome of the embryo. These cells now, I could make, and any competent stem cell guy can, I can make cells from everybody on this call, and in principle, I can make cells, same cells, from anyone in Ljubljana or probably anyone in Slovenia. And I am not exaggerating. To the best of my knowledge, there's never been a person who, scientists around the world who are making what we call iPS cells, or induced pluripotent stem cells, has not been able to make a cell line from a single person. Everybody who's been a donor for this, we've managed to do this. I was part of a program in California funded by the California Institute of Regenerative Medicine. And, there were groups in the US who made 9,000 cell lines from 3,000 donors. and all 3,000 cell lines, all 3,000 donors successfully made cell lines. So this is technology which has now become very, very simple and straightforward. It has advantages that you now can make cells which are genetically matched to the donor, which again, if you now want to go to therapy, you have a multitude of choices. If we take Parkinson's disease, for example, where you want to replace missing dopaminergic neurons, we can't get those cells from bone marrow or from any other adult tissue. We could make them from embryonic stem cell lines, but they will not be genetically matched to this guy. They'll be genetically matched to the embryo. But we could take skin cells from this guy, make iPS cells, turn them into his own dopaminergic neurons, transplant them without the need for immunosuppression. You know, the brain's a little bit unique in this regard, but we could similarly, from the same population of cells, we could make cells, as I showed you, from, you know, to repair his heart, to replace his lost beta cells in his pancreas. You know, we could make these intestinal gastroids that I told you about, which, oh, sorry, I didn't tell you about the gastroids. So that paper has just come out in Nature today. And again, a group in Cambridge in the UK figured out how to convert human embryonic stem cells into about day 18 to 20 human embryos, at least the gastrulation part of the embryo. So this is the developing enteric nervous system, the developing digestive system, but it is very, very, very in human disease, and nobody has been able to make these before this. We've been able to take embryos to about day 14, and then we stop. These guys, without making embryos, figured out how to take pre-existing embryonic stem cells and then turn them into early essentially gastrulation embryos. And so this would be very important for looking at diseases of the digestive system like Crohn's disease, inflammatory disease, and others. I mean, this is just a very small list of therapies for which we're either using cells from bone marrow, from adipose tissue, from cord blood, embryonic stem cells, or more recently from induced pluripotent stem cells to make a wide variety of tissues for direct cell replacement, or in the case of some disorders like osteoarthritis and rheumatoid arthritis, maybe just using MSCs to go in and modulate the immune response. So again, if you look at clinicaltrials.gov, type in your favorite disease, you will pull up thousands of clinical trials for all of these diseases and more. And so, you know, finally, somebody who's been in the regenerative medicine, you know, cell therapy, stem cell space for 25 years or more, almost 30 years now, people have always said, when's this field actually ever going to give us something? And already it is. I mean, we have late stage clinical trials of macular degeneration, which have shown efficacy. Parkinson's disease, 3 clinical trials will start at the end of this year, next year. Spinal cord injury, Geron has been working on this for many years. They have clinical data showing efficacy. Diabetes, clinical trials of replacement beta cells, the list just goes on and on. One point I do want to make, and this is something else that we've been working on for a long time in conjunction with most of the groups that are shown on this slide, starting in about 2011, these researchers, Carl June's group at Penn and Michelle Sandelin's group at Sloan Kettering, began to publish data showing that they could, in patients with really advanced lymphoma who had failed all conventional therapy, including many cases had failed a bone marrow transplant and were within weeks of dying, by removing their white blood cells from their blood, genetically engineering them to target their particular form of cancer. At least in the early days, about 90% of these patients went into complete and total remission. And that has been durable, and most of them, at least 50% of them, have survived. I mean, Emma now, I think she was transplanted in 2010, and she's still alive and shows no sign of her cancer coming back. This has fueled a real huge interest in using the immune system, not only in cancer. As you know, cancer therapy is generally fairly non-specific, using very high dose chemotherapy, radiation, surgery, and in some cases, you know, drugs which are really ineffective. There's now a huge interest in trying to retrain the immune system to target cancer. This is work, the stuff I'm showing you here, which we call CAR T therapy, has been most effective in blood-borne leukemias, blood malignancies, but it's now being targeted into solid tumors. And if this really works in solid tumors, it's a real huge game changer, and probably the most exciting thing that's happening in cancer therapy in the last 50 years. Not surprisingly, based on some of our conversations previously, the pharmaceutical industries and the investment community has piled in this in a huge way. Michelle and Rainer are two founders of a company that not only raised almost $1 billion on the stock market on the basis of phase 1 data, but then they were acquired by Celgene for something on the order of $13 billion. So pharma's investing in this in a huge way because this is really transformative therapy. Patients in which this therapy is effective, we think, could have complete and total durable remission from lymphoma and leukemia, which we only see with the best in bone marrow transplantation. And this, I think, has fueled a big resurgence in the field of immunology to see if we can start to do this for autoimmune disorders, as well as for solid tumors and other hematopoietic disorders. So most of my time these days is taken up by trying to help people identify emergent trends in technology that, at least in my mind, are going to have impact on healthcare, and ultimately an impact on healthcare is an impact on life. So, and aging and longevity. So these are in no particular order, and I won't talk about all of them. I'll talk about the ones that I think are the biggest and probably the ones that hopefully will make the most difference. The first one is what we call omics. And omics is omics is a set of technologies where, the first instance of this was really the Human Genome Project, when we thought if we sequenced the DNA, in the chromosomes, that was going to be a gold mine of information. And it was, to some extent. It gave us the book of letters of the human genome, but it doesn't tell us anything about paragraphs, structure. There's no translation. That took a whole bunch of other technologies to come along. And what we have now is, everybody wants to have their genome sequenced, and we think that's going to be really important. Because of high throughput sequencing, the cost and the speed of getting a genome from a single person has gone from, several $1 million in weeks to basically today you can probably get your genome sequenced for $500 and it will take less than a day. But but then you need more data. That's, like I said, that's just the structure. What you really then need to know is what does that mean? And so there's a whole nother omics called transcript omics, which basically is what are your genes actually making, and what are they making in what cells? Because not all cells express all the same genes. Obviously, you have liver-specific genes, heart-specific genes, brain-specific genes. So that begins to give you some functional data. Then you actually, each transcript has the potential to be alternatively spliced into a number of different proteins. So one gene does not equal one protein, right? So then there's a whole other field called proteomics, where you then begin to look at the proteomic structure. Because even if stuff's being made at the messenger RNA level, that doesn't mean that the amount of protein is equivalent across cells or tissues or even individuals, okay? And then it goes on. Then there's metabolomics. We've talked a little bit about metabolism, Aubrey, of course, quite extensively. So this gives us a gold mine of information because it tells us actually how all of this stuff is working functionally, not just at the genome transcriptome level, but actually physiologically relevant stuff. Then you start to pull in additional data, clinical data. So all the clinical data sets, all the imaging studies, your blood work, et cetera, et cetera. Over here, Martin's in the way, so I can't really see what's behind this. Can I move Martin? I wonder. Yeah, there we go. So now you got microbiomes, you got glycomics, right? Microbiome, I'll come to that in a moment. This is basically what are all the bugs that live in you and on you, and how do they influence life, right? And disease progression, disease sensitivity, and your response to therapy. Now, if you take one patient in their genome, that's a huge amount of data. You start to lay all these other platforms on top of it, you end up with massive, massive amounts of data, which absolutely are going to require machine learning, deep learning, or what I call synthetic intelligence to put all of this stuff together. And now, it's not enough to do this across one patient. In the UK, we have this 100,000 genome projects. In China, they have a 1 million genome project. There's not enough brain power, enough time for humans to take all of this data, let's say, even across 100,000 patients, all these different omics, and make any sense of it at all. This is where, really, really smart companies like, in silico and Aubrey already talked about Geonescence, and DeepMind, which is the world's leader, or was the world's leader in synthetic intelligence until very recently. We're going to need these immense, powerful computers that can learn how to play Go de novo and apply it to this. And I think once we do this in an unbiased, completely open-ended way, where we're not telling the machines how to read the data, we're just leaving them to data mine the data on their own without any preconceived ideas about what that data means or how to mine it. I think we're going to come out with stuff that we've never even anticipated. We're going to see connections between microbiomics and metabolomics and transcriptomics that we've never seen and we've never looked for. So I think this is really exciting, and I think this will address some really deep-seated problems and lack of understanding we have about, let's say, just around processes associated with aging and susceptibility to age-related disease, and the damage that is incurred in bodies over time when we can really dig down into these data sets. Gene editing using CRISPR and other gene editing technologies is huge. A lot of this is going to be done in treating disease, but a lot of it is also going to be used in agriculture. It's going to be used in designer animals, designer crops, potentially in the ag bio business, plant-based meat, fake meat, et cetera, et cetera. We haven't talked much about brain health. I'm a brain scientist predominantly. So pharmacological or technological enhancement of brain function. As I said, there's very powerful stem cell populations in human brain that give rise to new neurons every day, particularly in the hippocampus. We know nothing about what regulates this process. We know nothing about how to impact on it. And this is an area where pharmaceutical companies have been reluctant to jump in because it's very hard to understand how we're going to be able to analyze this in living human patients, even with the best imaging capability that we have. And then down at the bottom, I mean, this is a really interesting ethical conundrum, because if indeed I can make a pluripotent stem cell line from every single person, that means I can make cells of any specific lineage from them, which also means that I could make gametes from them, that is, eggs and sperm. And at least in principle, this works. We've made mice actually from mouse cells that make mouse sperm, they make mouse eggs, and they're functional. We've made mice from them. And there's no reason to suggest that we can't do this with humans. So you could have people who are completely infertile. We can make functional eggs and sperm from their pluripotent stem cells. It even raises the possibility that you have somebody who is no longer fertile, say 80 years old, who decides they want to have another child, and we make artificial eggs and sperm from them. There's a whole bunch of ethical issues about being able to use male cells to make both eggs and sperm from males. And so you could, in principle, perhaps have a baby with yourself, at least in the dish. And so my bioethics friends are really interested in this. And clearly, this raises a lot of regulatory issues about whether or not these cells should be used and how they should be used. Currently, it's restricted in the UK, at least, only to be able to use them in research. But there will be pressure, I'm sure, that these cells are able to be used in fertility treatment. So watch that space. I realize I have about 1 minute left. I just want to finish with a couple of things that I think are much further away. Some of this is really only in the last couple of years, basically reliant on our ability to do a lot of heavy AI data crunching. The big one for me is microbiomic diagnostics. That is looking at the populations of bugs and fungi that live in us and on us, particularly in our gut. There's a big paper that came out in Cell today that analyzed microbiomic impact of about 540 different drugs that are FDA approved, and shown that in many cases, depending on your microbiome, your body metabolizes those drugs either to something that has some toxic metabolites, or that the drug is wholly ineffective. That is, the microbiome basically metabolizes the drug in a way it has no clinical benefit. And we've sort of known this for a long time, but this is some of the first data that really shows this. And there's a whole bunch of stuff about microbiomes and people who are obese, who have type 2 diabetes versus those who are thin, disease susceptibility, clinical use of microbiomic stool replacement in patients with inflammatory bowel disease and Clostridium differens, which is a very, very nasty bacterial infection of the gut, which kills a lot of people. So, you know, there's a big interest in trying to look at the microbiome as the next diagnostic area that will be important in human health and disease. Some of these other ones, I'm going to sneeze, I think. Excuse me. Neuroprosthetics, you know, artificial limbs. This is advancing really, really rapidly. Similarly with exoskeletons. 3D tissue bioprinting is coming along. This is something I was really interested in doing at GE, and we made a lot of progress before it was abandoned. And the whole idea is to basically be able to create printed tissues. I mean, this is from Tony Atai's lab, who's one of the world's leader in this field. Basically, it's very simple. You image a structure that you want to make size specifically for your individual patient. You turn that structure into a 3D computer-assisted design program. It tells the printer which cells to place where in the mold. You put it in a bioreactor, you grow it up, and you basically then administer it to the patient. Right now, we're at the stage of being able to make some relatively simple tissues, things like ears, synovial tissues, menisci, things like that. But I don't think it will be too long before we start making some things like heart valves and maybe some more complex tissues. The printer technology is basically there. We just need to make it amenable to being using cells instead of inks. But the technology, for the most part, is there. Last but not least, this is one of my favorites. This is Eddie Chang, who's a neurosurgeon in San Francisco. And what Eddie's basically doing in live awake patients who are being treated for temporal lobe epilepsy, particularly in their left temporal lobe, which is where all the high real estate cognition areas are. Eddie is mapping out seizure areas so he can go in and safely remove them without damaging speech and motor strip and other important neurological functions. But at the same time, he's beginning to map out, in this case, how we actually process speech, which parts of our brain respond to which sounds. And then ultimately his idea is to turn it around and figure out how we create speech. And then with electronics and implantable devices, could you use that as a way of, in somebody who's had a stroke in their left temporal lobe and is aphasic and has lost the ability to speak, could you put a stimulator in another part of their brain and basically restore speech? So Eddie is one of the world's pioneers in electrocorticography and machine brain interfaces, and somebody I definitely keep an eye on. And if I had money to invest, I definitely would invest in this guy. So Martin, with that, I'll stop. I probably have gone over, but if there's time for questions, I'll happily take some.
Thank you, Stephen. It was a wonderful lecture, really interesting, and I think that the audience is happy to be a few minutes longer than official time, but it was really great, like the whole view of the field. So I will ask you now questions from the chat.
Sure.
So the first is from Didier. Do you think that we could have gene therapies for longevity in not so far future?
You know, gene therapy is really a one-target approach. You know, so a gene therapy, obviously for hemophilia, you're going into, you know, the hemoglobin genes. A gene therapy for spinal muscular atrophy, which has been approved. For example, you're trying to replace the SMA gene which is defective. So to have a gene therapy that treats all the myriad aspects of aging, no, I don't think so. I think that's much more complex therapy than gene therapy could deliver. Gene therapy at the moment really works on single gene disorders.
Okay.
Or single cell type disorders, because we use gene therapy in Parkinson's disease as well, for example.
Another question, don't you think that it is important to make human clinical tests for longevity for older people easier and faster?
Yeah, I mean, of course. Clinical trials the problem with clinical trials is they're very expensive. You know, as somebody who's done this, I mean, just a simple phase 1 trial where you're looking at, you know, does this drug, does this therapy, is it safe? You're not even looking at does it work. Efficacy is phase 2. So for those of you who don't know, clinical trials are in phase 1, phase 2, phase 3 generally. In phase 1, you treat a small number of people, maybe 12, with you treat 3 with one dose, 3 with another dose, you know, and a dose escalation. That simple trial of 12 patients generally will cost $1 million, depending on where you do the trial. A phase 2 trial where you're now looking at treating versus best of care or a placebo, those trials routinely will cost $5 million. So clinical trials are generally certainly phase 2 and on are generally done by pharma companies because they're the ones who have the budgets to do it. But yes, I mean, we need to do more clinical trials for more things. And certainly recruiting elderly people is definitely very important. Recruiting more Black people, recruiting more women, recruiting more children, you know, because most of those groups are excluded from clinical trials.